All fair points, and but I think Bruce Johnson would say very nicely, Jeff's boss, our former boss, would say that 100% of our benefit has helped 20% of our patients, and I think that is so true. But where I think we should be looking at is that how we can change this landscape prominently. And I think Tom made a very poignant point that while we can all be quote unquote excited about those impressive benefits of the EGFR TK inhibitors, try telling that to a 40-year-old never smoker with a 5-year-old kid, and I've done exactly that. And it's really not promising. And some of my non-oncology colleagues, physicians, look at this and really think we are out of our minds when we talk about this as significant progress. But I do think this is how you make steps, and we have been struggling with empiric therapy. Now it's going to be more science-guided therapy and rational therapies, and I think I am by nature slightly a realist with pessimistic overtones, and I have to confess that I've become a little bit more optimistic over the last few years. Not so much because this drug has come or that drug has come. I think our own approach is now changing, and we are going to unearth the basic biology of cancer better than we had ever done. So that's where my promise is. So I'll just say a few words before I go to that section of how we are going to make the promise. And the ALK positive, I'm sure everybody in the room knows about this. The EML4 ALK translocation happens in about 3% of patients with lung cancer, perhaps a little less if you take all comers, but about 3%, maybe more, right, for adenocarcinoma. And discovered somewhat serendipitously through a very laborious approach, and the paper was published about 6 years ago in Nature. But the beauty is that today you don't have to do those tedious exercises. You can do this through this large-scale genomic thing. I'll show that in a minute. So that opens up a lot of possibilities of how we can find these things in the coming years. But what is remarkable is that once you have this ALK rearrangement, the kinase that gets activated can be turned off, not just by one drug. Now about half a dozen of them are showing up, each one looking better than the previous one. So an entire field has opened up in the last 4 or 5 years when nothing existed before. So that's the stunning part. Look at the responses. They are quite dramatic. Sure, eventually they progress, but now we have better options for these patients. And this is a study that compared use of crizotinib directed against the ALK rearranged patients versus standard chemotherapy, showing that it was better. Again, these improvements are very modest, even though more so than when we compare chemotherapy one versus other, we still have a long way to go. And but these are, as I said, the early parts of it. Where we are really getting better, and I think we should get a lot better here, is understanding how the cancer cells evade the response to these agents and how we can target them. And this is a beautiful slide Ross Kamich showed a while ago, or published a while ago. And essentially, when these tumors with ALK rearranged non-small cell lung cancer begins to grow after exposure to crizotinib, they do so either using the ALK as a pathway or ALK kinase or ALK independent processes. And now there are different approaches targeting on what's happening in this. And sometimes these things actually undergo conversion, and where suddenly the ALK no longer becomes a dominant thing, EGFR mutation drives the cancer cells. This once again underscores the importance of rebiopsying these patients and studying the molecular profile of these patients to direct appropriate therapy. That's a new paradigm. That's something 5 years ago would not have been considered reasonable or even possible, but not today. A lot of us are doing that in our clinics to direct therapy. And there are a number of ALK inhibitors in development. You may not have seen this data unless you had been to ASCO or one of the ASCO meetings. And I'll just show the LDK378. This is a Novartis ALK inhibitor. This actually was, again, a large study, phase 1 study, looking at different populations, ALK-positive lung cancer and the ALK-positive other cancers. And by the way, a lot of tumors have ALK overexpression. That doesn't necessarily mean this ALK is rearranged. This is a situation where the EML4 and ALK in lung cancer come together because of inversion in chromosome 2, where these things get flipped and there is a kinase activation there. And one group of patients had prior therapy with crizotinib. The other one did not have crizotinib. And if you look at the results, look at the bottom number. That's the most important. About little over 50% responded very nicely to therapy. And it didn't matter whether they got previous crizotinib or not. In fact, patients who progressed on crizotinib still responded very well to this agent. And we have seen some early data which have not yet been published with other agents that look actually quite promising as well. And if you look at the waterfall plot here, pretty much everybody responded except a few, and it didn't really matter whether they got crizotinib or pre-crizotinib. And the side effects are reasonable, fairly well tolerated, not a problem. And there are now a couple of major trials ongoing looking at this, both in patients who have been exposed to crizotinib and those who have not been treated with crizotinib, to see whether this would be better than crizotinib in the frontline setting, or can this work in crizotinib. And I'm reasonably certain that this drug is going to, if it continues this trajectory, this will be an option for our patients fairly soon. And then the issue really comes, which one of those 6 or 5 ALK inhibitors will take place in the salvage setting, and then we'll be having this debate. Should we use LDK-378 in the frontline setting or crizotinib? And some of these agents, actually I should say many of these agents cross blood-brain barrier. There have been some spectacular responses in the brain tumors in these patients, so this is certainly a very promising area. My own personal view is that the field is moving faster in the ALK rearranged non-small cell lung cancer than in EGFR mutant non-small cell lung cancer. We now have, I think we are beginning to see the early days of fairly reasonably active therapies, even in patients who progress through frontline ALK inhibitors. So that we cannot say, unfortunately, with EGFR mutant non-small cell lung cancer. It's a bit of a shame because the EGFR mutation is far more common than the ALK rearrangement in non-small cell lung cancer. Progress nonetheless. Second is BRAF. BRAF again illustrates an example of how common genes can be altered in many cancers. And BRAF mutations, there are different genotypes, and V600E is a fairly common thing. As many of you know, it's a very important driver in melanoma. And dabrafenib shows activity in melanoma. These are the data from melanoma phase 3 studies. Once again, looking very much like the ALK rearranged non-small cell lung cancer with crizotinib, or EGFR mutant lung cancer with erlotinib, or afatinib for that matter. And so now this is this ASCO, there's a large international study. And the group came together to study this. One is in the US as a part of Lung Cancer Mutation Consortium, where patients with BRAF mutation with lung cancer got dabrafenib. And this small study, but again, these are not very common mutations, and you can see a response rate of 40%. This is actually a bit of a relief because in colon cancer, this is not an active agent, at least from what we know. So clearly in lung cancer, the fact that it works in that small number of patients is actually a bit of a relief. And it's too early to say how long the duration is, and you can see that some of the patients have gone on, some even gone on past a year. So too early to say the median duration of response and how often these things will last. But clearly this seems to be the BRAF inhibitors now show some promise in this population. Small number, 1% or less than that, but still it's an active agent. And again, you can see the nice responses on that liver as well as in the lesion in the hilum. And you can see that. Of course, we covered PD-1, so I'm not going to. I'm going to skip that. But that's a new agent as well. So I'll make a point that that's active as well in this group. And so I can give you these 3 examples. But if you ask me, wait a minute, if this is the way we're going to make progress, these are 1%, 3%, how are we going to make any progress? And so I just want to, some of you may have seen these slides if you had been to some of my ASCO talks, and of course, most of the faculty have seen this. And so just so that others get a feel of what's happening, I thought I'll show some of these slides. The important point is that there is no one single way a cell can become cancer. There are many ways a cell can become a cancer cell. When we talk about mutations, we commonly talk about a point mutation where you can say a single amino acid is replaced and that changes the conformation of the protein and directs the gene to be more active in case of oncogene, or loss of a tumor suppressor. But you can have copy number alterations, you can have structural rearrangements, as you see. A classic example is the BCR-ABL EML4 ALK, and there are inversion. Inversion is a great example. EML4 ALK is a great example of inversion. And then you can have a higher expression of the genes, and all of them can do that. So that's one problem. The second problem is that this is the work that we did as a part of the Cancer Genome Atlas project for squamous cell lung cancer. I showed that at last year's ASCO when we published this in Nature last year. The Cancer Genome Atlas is a very ambitious project. In terms of cancer biology and cancer or medicine, this is, in my opinion, equivalent of going to moon. And I don't mean to exaggerate the importance too much, but this is for the first time a systematic attempt was made where we wanted to study all the 20,000 genes in the cells. And remember, until about 10 years ago, we did not even know how many genes we had in each and every cell. And thanks to the Human Genome Project, where a pooled human genome from 50 individuals, but mostly came from a few, and were actually pulled together. And this, for the first time, the whole human genome was mapped. This is the massive effort undertaken by 4 major institutions, including the one I come from in St. Louis, and Broad, and one in Texas, and one in UK. All 4 of them and a number of other institutions joined together. And the cost of doing the Human Genome Project for sequencing one pooled human genome was about $3 billion, B as in boy, $3 billion. It took 10 years to do that. But out of that, a lot of things were developed. The roadmap was created, the software to analyze them actually was developed. The computing power improved tremendously during that time. And also the technologies improved where you can start sequencing. And about 4 years ago, my colleague Tim Lay at Washington University and Rick Wilson and Elaine Mardis, they sequenced for the first time a whole genome of a cancer patient. Again, that project took about a year and a half for us to do. One patient with leukemia. And these results were published in Nature. And that actually, whenever you do the cancer genome, you actually do 2 genomes, one from the tumor DNA, one from the patient's germline, essentially to ask a question, what genes are altered in the tumor tissue compared to the genes that you're born with? So you do 2 genomes together, and the 2 genomes together cost about $1.5 million. So far cry from 3 billion to $1.5 million. And in the last 4 years, now we can do a whole genome, paired whole genome, in literally 6 weeks. The shortest we have done is got the sample, done the sequencing, done the analysis, all in 6 weeks, and published 2 papers 2 years ago, and all for about $20,000. That's all for discovery. I'm not proposing that we do in the clinic. We have done that in the clinic just to show that we can do it and change treatment. But most of the work is done in discovery. So back to the TCGA project. The TCGA project is a $300 million project covering about 20 common diseases run by the United States government, thanks to the taxpayers' dollars, and where the genomes of common and some uncommon diseases are going to be mapped. The first phase of TCGA will be wound down by next year. And by then, we have studied the map of glioma, colon cancer, breast cancer, lung squamous cell, lung adenocarcinoma, and of course, a bunch of other cancers in the next year or so. And so we've learned a lot of things. And the squamous cell paper, we showed that, for example, CDKN2A is very common. That gene is lost in more than 2/3 of the patients. But look at how this is lost. It's lost in so many different ways. And you can see that it is mutated in a small number, and it's methylated or silenced in some. It is deleted in others, and they are, for most part, they are non-overlapping. In one patient, it was lost through rearrangement. We had to pick up through the whole genome exercise, and showing that a common gene can be altered in many different ways, and it may biologically may exert the same effect, but the treatment approach may be a little bit more different, and that's something we have to come to grasp this and study this. So when we did this studies, very quickly we figured out that if you look at the mutation per megabase of the genome, and that's highest in lung cancer and melanoma. That's what I was referring to a while ago. And acute myeloid leukemia, only 0.8 mutation per megabase. So acute myeloid leukemia is not a simple disease. It kills people. And whereas lung cancer, it's actually quite dramatic. And most of these mutations actually are not driver mutations. They are passenger mutations. What do we mean by passenger mutations? They are there. They have been acquired over the life of a cell. They are sitting there. They probably don't contribute to the disease process, but they do clutter up our interpretation. So when we suddenly see, when we do this sequencing, I'll show you in a minute, this is actually the picture of a whole genome landscape of a patient of mine. This is done through whole genome. Just out of curiosity, aside the faculty members, anybody has seen this kind of a picture? Do you know what that means? Do you know what this picture is? I'm just curious. I've shown this picture one too many times. I just want to make sure that I'm not showing this. So any of you have seen this in the audience? Okay. These are called circos plots. This is actually a neat way. If only our last speaker has not stolen the PowerPoint or this laser. Is that you?
No, I was there.
Oh, it's a blind speaker here. So this is actually a very neat way of describing a genome. And bear with me for a minute. You'll just find this interesting. And this is done when we do what is called the whole genome analysis. Whole genome means you look at the coding and non-coding regions. Exome sequencing, look at the coding regions. Those are the two types of DNA sequencing. And of course, you can do the limited panels like EGFR or a 20-gene panel and other things. So when we look at these whole genome, we look at all 20,000 genes. We look at, as I said, coding and non-coding regions. This is the most intensive, labor-intensive work. The other sequencing is transcriptome sequencing, where we look at the RNA sequencing. That's also becoming very, very useful. Through that approach, now we can find all kinds of fusion genes like EML4 very readily within a matter of 6 weeks in any given tumor. And so this is called the Circos plots. It's very neat. And it goes around the circle. Each one represents one chromosome. This is chromosome 2, 3, 4, etc., on and on. So each of these labels, if you come closer, you will see all those letters, or mutations in that single nucleotide variations or mutations in the common parlance in each of this chromosome region. And these are copy number changes in the segment. These are structural rearrangements within the chromosome and in between the chromosome that you can see the lines crisscrossing. So this is a patient with lung cancer. I show these two slides. We published this paper last year. But I think these two, I'll show you the next one in a minute, they actually nicely demonstrate what happens in lung cancer patient. Adenocarcinoma stage 1 or 2, I don't remember, and had 50,000 mutations in one tumor. And you may, 20,000 genes, but how do you have 50,000 mutations? Same gene get hit many times sometimes. And of course, most of those mutations probably don't play a role. But the problem is when you study these genomes, if this patient has EGFR mutation, it's easy to pull out and say this is what's driving the cancer. But if you don't know, if they are looking for a new genes that are altered, it's very hard to sift that out of this mess with 50,000 mutations. That becomes harder. But then if you look at the never smoker with lung cancer, adenocarcinoma, it looks very different. Very few mutations, very few structural rearrangements, copy number alterations, and that's what we see repeatedly. A tenfold decrease in the mutation burden in never smokers. And we are using this. We are now doing a 100-patient study of never smoker to see in the whole genome exercise how we can figure out these targets. And I personally think the biological processes are somewhat overlapping, and we'll be able to sift through the clutter or look for the needles among this huge haystack in these patients better than the previous patient I showed you. So the mutational burden is very high. But there is also an opportunity here. And with the better bioinformatics, we are able to understand this better, and the bioinformatic tools are getting better and better, literally month by month. And we are also, when the tumor is mutated like this, and what happens is that there are a lot of antigens that could potentially be expressed on the cell surface, and these patients may be prime candidates for immunotherapy through all kinds of novel approaches. So in fact, in the immunotherapy world, when the genomics and the immunotherapists get together, these are the patients that they get excited about, not these patients, and they are unlikely to have antigens that they could go after. So there is a potential value, and I personally think eventually we have to employ a combination in some sort of a sequence, chemotherapy, targeted therapy, and immunotherapy, and also respond to the dynamic changes in the genomic landscape. While I show you this picture, when we repeat a biopsy after some kind of a targeted therapy, I bet this would change quite a bit. And actually, I did not bet. We see that actually, and the genomic landscape is changing. Remember, this is an evolution that's happening in a very small compressed period of time. And in the squamous cell paper, we did show that nearly 2/3 of the patients with squamous cell lung cancer have targetable alterations. In fact, the most important one is the PA3 kinase PTEN pathway, and also the RTK pathway, as we see here, various members of this pathway. For example, the TSC1, TSC2 tumor suppressors. There was a paper recently in bladder cancer showing that everolimus is very sensitive, or the tumors with TSC1, TSC2 alterations are sensitive to everolimus. So we need to study these things as we go along. And this is actually a paper currently under review. This is again from the TCGA work, and on adenocarcinoma, we are going to show some novel mutations in these tumors in the upcoming paper. And this is the work from Matthew Myerson showing that, again, we should be looking at epigenetic changes. And we see that not only Matthew's group, our own group has seen many times these epigenetic changes in lung cancer. And we need to really understand how best to understand and target them in the coming year. So that's going to be a major thing. The good thing about the Cancer Genome Atlas is that this information is publicly available, and the busy clinicians obviously won't need to go there, but this will be available for investigators from around the world, and several groups are mining this data and publish papers on that or validate whatever they find in the lab, and I won't go into the details, but already there is this new fusion with FGFR that has been reported by a group from Michigan, again validated using TCGA samples available in the public domain. So I think we'll see a lot more of that. By the end of this year, we'll be analyzing all the 1,000 samples we've collected from the TCGA and be looking at the genomic changes on all 1,000, and we'll be publishing another large paper outlining those changes together. And when we do all these things, we find that there are a lot of other proteins that are altered. ROS1 is a classic example. If you look at ROS1 is very closely related to ALK. So not surprisingly, when you give crizotinib, these patients have very nice response. Again, small number of patients, literally everybody seems to respond here, at least to some extent, although the overall response rates are around 50 to 60%, as you can see here. And this is one of the patients from Alice Shaw showing a very nice response before and after therapy with crizotinib and with ALK rearranged non-small cell lung cancer. And these tests are available in many centers, and if anybody wants to do it, I'm sure you can find that either in your lab or our center does it. I'm sure a lot of other centers close by will do this on a routine basis. And this is an example in our patients, in our study of patients with lung cancer. When we did a whole genome analysis and transcriptome, we found actually more than 60 fusion genes in these tumors, and each one is unique, not found again, and many of them involve metabolic enzymes. And we are beginning to study these things systematically, and a lot of we look, I don't know whether we really see what we have to see, and or I should say the other way around. And clearly we need to study these things in the coming years to see how best we can target them for therapy. But one thing is clear, we don't have to do the laborious studies that had to be done in the past to look for the novel fusion genes, and now it's becoming an automatic process, and we and others are pursuing them. Not to depress you, but most of the studies, including the TCGA work, is done essentially focusing on the 2% of the genome. That's the protein coding region. Although few of us have done some whole genome studies, TCGA for most part is focusing on the coding regions. And but I do think a lot of information is buried in this non-coding regions, and particularly there's a lot of interest both from our group and some other group to look at the non-coding RNAs, long non-coding RNAs and link RNAs, and how they can alter. Again, the theory is that can we find master regulators that affect multiple genes? In other words, is there a way we can disrupt the master regulators or the ringleaders, so to speak, and thereby we can make dramatic progress in these patients. And finally, I think you have to keep in mind that these tumors, as I said, are not static. They evolve continuously. And now with all the deep digital sequencing, we can actually estimate what proportion of your cells have this mutation or the variant allele that we can talk about. And if you think that the mutations are present in all the cells, since most of them are heterozygous, and a 50% variant allele frequency means you have one single cluster. But then here you can see that three different frequencies, three different clusters, that means there are three different clones. So this way we can look at the clonality, and through that we can find how many different types are there. This is a beautiful work done by Charlie Swanton from UK, published last year on kidney cancer, but the lessons are similar for other cancers as well. And where his group took a kidney cancer tumor, multiple sections were done, and you can see that there are tumors, there are genes that are mutated, that are common between primary and metastatic, primary from different sites that are shared, some are unique and private. And what probably happens is that there are some small clones that are in the primary that acquire some mutations, and they then become metastatic. And then at the metastatic disease side, they evolve further, and that's where they have those private mutations. And so you can look at this. This is the branching tree model. And so you can see that the primary tumor, whatever caused the disease, probably started here. In lung cancer, this will be EGFR mutation. And then as it evolves, it picks up more additional mutations. And perhaps this mutation is the one that confers the propensity to brain metastasis. Maybe this one gives propensity for liver metastasis. So depending on which clones acquire what mutation, so these tumors evolve in different direction. So this is why, when we rebiopsy, this never goes away, because this is in the truncal event present in what we call the founder clones, and that are present, and that of course can give growth advantage if it is not suppressed. So you may say, wait a minute, just leave us alone. We would rather go take a walk down the Park Avenue. Forget it. This is not going to be fixable. Some days I feel like that, but some days I think we can probably get our hands around that if you are very patient, if you're organized and systematic. And this is the work done by Tim Lay in leukemia from our group. And he actually did a very neat study. And of course, in leukemia, they can do this, and not easy in lung cancer. So they did serial bone marrow biopsies at diagnosis, after induction therapy, and then eventually over time. And essentially, if you can see that by using this variant allele frequency, you can find different clones. And essentially, these are the founding clones, the gray bars. And then this tumor picks up multiple clones. And you can cytoreduce them. A few clones persist. And then at the time of relapse, these things expand, but then a new clone arrives, and that eventually overruns everything else and kills the patient. So the question really is, will tumors evolve in a predictable fashion? So in other words, you can actually prepare yourself for this resistant clone eventually would kill, or is it not predictable? So it's going to be very stochastic, and it is almost impossible. It can be many different ways these tumors can evolve. If that is the answer, then that's a bad news. And but even then, we may have to find repeat biopsies to study them and understand them. And perhaps a given tumor can only evolve in so many ways, or even more importantly, and I'm not sure whether that's what's happening, this clone is probably there very much at diagnosis, but our techniques are not so sensitive to pick them up. And sometimes people forget this. It's not as simple as running a flow cell and pick up the mutation and then read them off. You have to do a lot of bioinformatics and filter a lot of these things out. So there's a lot of science that is evolving going in bioinformatics, and our techniques are getting better and better every day, literally. And so with that, I think it's very possible that we may be picking up these clones that may be present in a small number, but suddenly have significant growth advantage over time. And I think that'll be good. I think the very, very poignant thing would be that Charlie Swanton actually made this observation that if you look at the tumors, how they evolve from the kidney cancer work, it's very similar to what Charles Darwin wrote many years ago, and how the evolution happened. So once again, the tumors evolved very similar to this. It's called the Darwinian evolution, and happening so quickly. And I think that's the big news from this. And finally, I think this is a beautiful slide that illustrates the problem, once again, with due credit to Charlie Swanton. And some tumors are like this, the palm tree you see. They just evolve, they become cancer, and they only differentiate or degenerate in so many ways. So you can easily take care of them. That may even be in some lung cancer patients. Maybe the ALK positive, ALK rearranged non-small cell lung cancer is like that. So you may be able to take away a couple of clones, and you probably may top it off with some immunotherapy or some kind of chemotherapy in some fashion, and you may be able to eradicate them, may be able to cure them. The chestnut tree type model may be a little bit more complicated. But you may be able to understand them by doing the serial evolution, and may be able to control them over time. And then, of course, there are really wild type that may take many years for us to wrap our arms around. So this is not going to be easy, and genome sequencing will not cure cancer. Genome sequencing will only tell us how complicated this business is and how foolish we were in trying to make advances by empirically combining therapies without any biological basis. By studying those key alterations and studying them dynamically and combining them in some fashion, I'm actually very optimistic. Did I say mildly? But now I'm very optimistic after listening to my own talk that I think we are going to make progress much more so than we did in the last 15 years or so. Thank you so much.